We're going to get this show on the road. Looks like we got a few people in here live. And if you're watching after the live show, hello from YouTube land. This is our third webinar so far in this little summer webinar series excursion from myself. I'm Richard. I'm one of the co-founders at Escarpment. Do a lot of different things here, including help direct our scientific program, yeast research and development, as well as quality control, all sorts of that fun stuff. And I'm really excited for this one today because I get to talk about Brettanomyces, which is one of my favorite topics in brewing. I think, Brett beer has kind of had its ups and downs. I think the peak of the excitement was probably a little while ago now, maybe 6 or 7 or 8 years ago. But Brett has remained relevant. It's a really cool yeast. We're going to get into why it's important, why it's interesting, and hopefully, why you as a brewer might continue to play around with Brett and unlock some of its potential, because it's really, really cool yeast. There's been a lot of yeast development in the other side of things with regular Saccharomyces yeast and developing it to make different flavors. But at the end of the day, Brett is still our flavor powerhouse in a lot of ways. So we're going to dive into not only some of just what we know about Brett, but also some of the more recent scientific information out there, including some stuff that we've done internally at Escarpment Labs to understand our funky friend Brett a little bit better. All right, so let's jump into it. If you're just tuning in for the first time, just a little overview of where we're at with Escarpment Labs. We're trying to make fermentation easier, based in Canada, but we work with breweries all over the world at this point, primarily producing liquid yeast, but we do have a dry yeast strain, the House Ale, ELD1. You can check out my webinar from last week about that yeast and how that was developed, what it can do for you. And we've got a whole team that helps make that happen, which I think is really important. It's awesome to be able to work with people that are so passionate about beer and fermentation every single day. And it makes us keep getting better at what we do, so we can keep giving you better yeast so you can make better beer, and ultimately just raise the quality of craft beer and ensure that every craft beer consumer, whether that's at home or whether that's in the tap room, gets a good experience and leaves feeling excited and inspired about craft beer. Okay, so Brett, why do we bother? And for those of you live, I want you to sound off in the comments. Why are you interested in Brett? Why are you here? I'm curious, legitimately curious to find out. This is also a good test to make sure that the comment section is working. I'm going to give you like a minute to answer that question. In the meantime, one of my favorite things about Brett is just looking at it under the microscope. Cells are very different shapes from, regular yeast cells. Normally, Saccharomyces yeast does what's called multipolar budding, so it buds basically wherever it can on the cell. Brett has a different way of budding, so you can actually see these little buds in this picture in the background. It does what's called apiculate budding, meaning that it buds at either end. So under the microscope, Brett is fundamentally different from a irregular yeast, so it's pretty easy to tell them apart. And that's something you want to look for if you want to make sure that your Brett is Brett, or if you suspect that your beer has a Brett contamination. You can look at the yeast under the microscope if you have a microscope, and if it sort of has that lemon or sausage shape, then usually that indicates that it's Brett, versus the Saccharomyces that will be more sort of round, and it won't have like any sort of tapered ends. So just a key difference. Brett can get really wild too. It can make what's called pseudohyphae. It can make really elongated cells that almost form like a web, almost like the mycelium of a mushroom. So they can do that as well. So you can get some really, really cool looking cells. So why are we here? I got some answers in the comments. This one from Jeffrey says, I think the stuff we can make is more interesting, which I think is true. You can make some really interesting beers with Brett, for better or for worse. Another guy just says Belgian brewer, which says it all. Mark says for the funk, which is, there it is. Yeah, Brett brings the funk. Caleb says mixed firm beers are some of my favorites, especially Lambic and Gueuze. So those beers in the Belgian tradition. And Vitor says, let's make more funk. But Jeffrey clarifies, don't want barnyard or wet dog though. So keep them coming. That's awesome. That's why we're here. We're here for the funk. We're here for the flavor without getting things too off the rails. That's really what we're here for with Brett. That's the one thing that Brett offers us as brewers, is that it offers really diverse and interesting flavors. There's flavors that you will get from Brett Brettanomyces that you won't get from your regular run-of-the-mill Saccharomyces yeast. Some of those are positive. Some of those are positive in certain contexts. Some of those are potentially negative. So it's important to keep that in mind. There's a lot that it can do. Every icon is there for a reason, because every icon represents a flavor that someone at some point doing sensory analysis at Escarpment Labs has identified in a Brett beer. So that could be anything from goat cheese to bacon to pineapple. So like, Hawaiian pizza right there, I guess. Mango, horse blanket, urinal cake, strawberry, apple, Band-Aid, bubblegum, barnyard, Parmesan, clove, pear. It can do it all. There's a lot there. And what's really cool is that you can select specific Brett strains to drive specific flavor profiles. That's something we find is some strains will make more pineapple than others, for example, or some might make more strawberry or cherry. So it is really something you can do is select specific Brett strains for specific flavor profiles. So, for example, if you want to push that pineapple, you can pick a strain like Brett D that really is very pineapple forward. Or if you want to push the citrus, you can pick the Brett C or the Berliner Weisse strains, which we'll talk about a little bit later. So yeah, this might look familiar to people who saw the first webinar about our yeast flavor and biotransformation guide. Really, this is just showing how Brett flexes on Saccharomyces yeast, because it is a flavor powerhouse. It produces flavors typically in much greater abundance than Saccharomyces yeast does, especially when it's on its own in a primary fermentation and not in a mixed fermentation, although it still does produce quite intense flavors in a mixed fermentation as well. However, it's harder to control this. One of the trade-offs with Brett is that it's always producing some of these flavor compounds that might be considered distracting, like these what we call volatile phenols. And in the case of Brett, it's not just the clove that regular yeast makes. You also might get a smoky bacon, sort of wood smoke. You might get that like barnyard funk. You might even get something that's perceived as like solventy from the phenolics of Brett. So that's always in there, balancing out with the fruit. So that's something to really keep in mind when you're using Brett, is that, yes, you can get a really heavy amount of citrus, a really heavy amount of tropical fruit, but you might also have some of those sort of smoky or barnyardy phenols that balance that out. So if we're looking at like what does Brett make in beer, I just wanted to highlight some of the key compounds. So we've talked a little bit about general yeast flavor, but we didn't really have an opportunity to dive in specifically with Brett. So when we're looking at Brett, really the primary flavors we're looking at in terms of what it makes are esters and phenols. So the esters, we have quite a big range. I'm just going to highlight a few. Ethyl acetate is one that is pretty common with Brett. It gives you that sort of pear kind of characteristic. It can be unpleasant if it's at high levels, and it is possible for this ester to be present at really high levels in Brett beers, especially if those beers have acetic acid or are exposed to oxygen, so that acetic acid or ethyl acetate is formed by the yeast. Brett does have the ability to make acetic acid from oxygen and alcohol. So if your Brett beer gets exposed to too much oxygen, it can go vinegary. But at low levels, this is a nice flavor compound that adds complexity. So that's one of these ethyl esters, for example. On the top left, a lot of those alcohols and other esters, it actually reduces. So the isoamyl acetate, the banana, and the phenylethyl acetate, those are esters that are produced in abundance by some Saccharomyces strains. Brett actually will typically reduce those into the equivalent alcohol, so phenethyl alcohol and isoamyl alcohol, which have a lower, a higher sensory threshold. So in practical sense, typically if you take a beer that has a lot of that sort of banana aroma and pitch Brett's going to reduce that banana and probably replace it with some other flavors. So in terms of what Brett's producing itself, you get a lot of the fatty acid esters. ethyl hexanoate, which is one of our favorites. That's that pineapple tropical fruit note. You might get that from some Kvike yeast. You also will get that in abundance from Brett's, especially Brett D. Ethyl octanoate, pretty similar. Same thing with ethyl decanoate. I find that they just get a little bit more apple-like, apple or plum-like, as the carbon chains get bigger. They're all pretty similar molecules. But all of these contribute to the fruitiness of Brett. And so does biotransformation. So Brett is quite capable of biotransformation. One example is that it can produce citronellol, beta-citronellol from geraniol in hops. So it can turn that floral aroma into a citrusy lemongrass kind of aroma. Brett also has often much stronger what's called beta-glucosidase enzymes. So it can take flavorless precursors in your hops or even fruit, other ingredients. It can use this beta-glucosidase enzyme to break up a bond and release that flavor-active compound. So it tends to have much better ability to use this type of enzyme than Saccharomyces does. And some Brett strains are much more active than others as well about how they use beta-glucosidase to release aromas. So interestingly enough, the Brett strains that are weaker fermenters are often better at the beta-glucosidase enzyme. So you can always look to something like Brett C as the classic example. That's a yeast that has pretty high beta-glucosidase activity, but it's a pretty poor fermenter. So there's also opportunity to combine Bretts. So you can, for example, combine Brett C and Brett D, get some of that beta-glucosidase activity and release some aromas from the hops. Often you do get some more free terpenes. So you might get, or terpene esters. So that often comes across to me as more citrusy or more like mango-like. Plus, if you have something like a Brett D that's making ethyl hexanoate, you're going to get more of that pineapple tropical fruit. So if you're looking to push that tropical profile, there's some cool things you can do with blending Brett strains. And then we get to the funk. So I sort of just called this ethyl phenols. There's a few different ones. There's 4-ethyl phenol and 4-ethyl guaiacol, and some minor ones. But this is what, when we're talking about the funk, this is mostly what we're talking about. These are these phenolic compounds that have been transformed from flavorless precursors in the malt by the Brett. And these are the compounds that are perceived to us as that sort of, I say funk, but really the components of funk are like the smoke, the barnyard, the clove, maybe the solventy band-aidy kind of thing. There are other contributors to funk, like Brett does produce some organic acids. So the precursors of those ethyl esters are organic acids that come across kind of like goat cheese or goaty, and that certainly does contribute to the funk. Some Brett strains make more of those organic acids than others as well. So that was just a pretty deep dive into flavor chemistry of Brett. I'm just going to talk a little bit about what you can expect when you're fermenting with Brett. If you are curious to learn more, we do have some older webinar recordings up on our YouTube channel, so you can go and check that out if you want to learn more about sort of the context of what we're talking about with Brett. We're kind of diving into the deep end here a little bit, but I'm gonna try to ease you in. One of the trade-offs with Brett is that the fermentation is a lot slower and a lot less reliable than a normal, regular yeast fermentation. So if we look at a typical fermentation curve, for example, Brett has a long lag time. It can often lag 3 or 4 days before it does anything. And then if it's on its own without any other Saccharomyces yeast in the mix, it tends to have a very linear fermentation curve, or line in this case, where it will just sort of just start chugging away at fermenting the wort, but never really hit an exponential fermentation in the way that a lot of Saccharomyces does. Whereas, your Saccharomyces here is probably done around day 7. Your Brett's going to probably take 3 or 4 weeks to finish out fermentation if it's on its own. And that's even true if it's in a co-pitch with the Saccharomyces, or if it's added after the Saccharomyces. It's going to take the Brett usually a few weeks to express itself and ferment. And why is this? Part of it is just because Brett responds to its environment differently than our normal Saccharomyces does. Regular Saccharomyces yeast is really easy to use because it's really compatible with a lot of things we're doing in brewing. Brett is a little bit trickier because it has a bit of a stranger metabolism. So One example here is what's called the Custer's effect, which is the central struggle of our funky friend. It's basically where if Brett um, is shifted from an aerobic environment to an anaerobic environment, it tends to have an extended lag phase. And when we're looking at the brewing process, this is a shift that we're putting the yeast through, and there's a reason why we do this. Um, Typically, in a brewing process, we're going to make a beer wort, an unfermented beer, and we're going to pitch yeast and we're going to oxygenate because that oxygen is going to help the yeast grow, which is true for Brett as well. If you feed it oxygen, it's going to grow faster than if it's fed just sugar, for example. So it will be able to produce biomass a lot more effectively in the presence of oxygen. But in a beer fermentation, you usually have this aerobic phase where the yeast is multiplying and they're picking up all of the oxygen from the environment to make more cells. And then they go through the adaptation to the anaerobic phase. And in the case of Brett, there tends to be a lag phase. They tend to have to reconfigure to this environment. And it can often take a little while before they start adapting and start producing alcohol, for example. So you can see in that lag phase, there's no alcohol being produced. The reason why we would never go like full aerobic on a Brett ferment is that we would also be full acetic acid at that point and basically making vinegar. So you don't really want to go there. So this is the central struggle. We'll refer back to the Custer's effect a few times in this talk because this is the thing we're trying to really overcome in a lot of cases with Brett, because if you can overcome it, you can make it ferment better. And the goal would be, long term, can we make Brett ferment at a similar rate as a Saccharomyces strain so that it's relatively similar to use, because that's not always the case. And if it is the case, sometimes it's not always consistent. So why does this happen? Why does Brett do this, but Saccharomyces doesn't? Like, they're able to kind of make that shift pretty easily. Part of it is just basic metabolism of the yeast. It's wired completely differently, and we're going to see why these two yeasts are so different in the next slide or two. But Brett, for one example, again, we're getting into kind of like hardcore metabolism here. We're going to zoom back to some practical stuff shortly. Brett doesn't make glycerol, basically. So that's one thing. Ordinarily, in regular yeast, they produce glycerol, which can be used to regenerate this thing called NADH or NAD+. They're generating NAD+. This thing's really important. This cofactor is really important for enzymes because it's required in a lot of the enzymes used to ferment sugars anaerobically without oxygen. So that means that Brett doesn't have an easy way to generate this cofactor. So they tend to be pretty slow at adapting to anaerobic fermentation. The way that Brett has overcome this is it has a neat trick. It imports nitrate into the cell. So it can get nitrate from the environment. It might get it from malt or hops or grapes, for example. And it can use that instead to generate the NAD+ that it needs to be able to ferment the sugars anaerobically. So kind of a different way of getting the job done is how I would describe it compared to Saccharomyces. And yeah, if you don't understand redox, that's fine, because I feel like I still struggle with it, and I've been a yeast scientist for more than a decade now. So why is Brett so different? Part of it is just evolution. These things are actually pretty far apart on the evolutionary tree. So this is like on the left, a simplified evolutionary tree of yeasts. Brett and Saccharomyces actually split apart about 200 million years ago. So 200 million years ago, there were still dinosaurs roaming the earth. Like, it was pretty different. However, since that time, both yeasts have become highly adapted to human fermentation niches, weirdly enough. This is what's called parallel evolution. They both figured out how to do the same thing, even though they're completely different organisms. It's like how life keeps evolving into crabs, if you've seen those memes. Yeasts keep evolving to figure out how to ferment wine and beer because it's advantageous, because humans keep making a ton of wine and beer, right? And then there's also keeping in mind that these are like 200 million years apart, like very, very ancient divergence, we also, can't really expect Brett to behave the same way that our Saccharomyces does. So that puts us at a disadvantage when we're growing yeast or fermenting with yeast or doing yeast research, because the rules that we apply to Saccharomyces often don't apply to Brett because it has a completely different genetic composition, right? It has different genes, different structure. It gets jobs done in different ways. Okay, so with that sort of intro, I'm going to get into some of the projects we've been working on with Brett. So the first one I'm going to talk about was Brettanomyces from Berliner Weisse, which is a cool topic. Berliner Weisse, especially, well, until the 80s and 90s, pretty much always had Brett. It was a Brett beer. It was a mixed fermentation beer, really special beer. These beers are why it was called the Champagne of the North, because these were beers that really had that champagne-like quality. They were dry, they were acidic, they were very carbonated, very light-bodied and easy to drink. So the like heavily acidic kettle soured stuff with no Brett isn't really Berliner Weisse, at least in the historical context. They're almost completely different beers. And turns out, and we'll get into this, the Berliner Weisse brewing culture had some pretty unique strains and pretty unique approaches to fermentation. So here's what some of them look like. We've been really fortunate to work with some Berliner Weisse nerds over the years and collect some really old bottles and open them up and taste them and save the dregs and then try to hunt for yeast. In some cases, these bottles are 40 years old, for example. There's some younger, some older, but over the years, we've been able to get a pretty good sampling of the historical Berliner Weisses that were out there and on the market, especially if you look at the sort of 70s, 80s, 90s in Berlin. And from a number of different breweries, although there was some consolidation later on as well. But really cool. It's fun tasting old beer. It's usually not delicious. If I remember from this tasting, most of them were like very oxidized, very cardboardy. But you can still sort of get the essence of, this was a sour beer, this was a mixed ferm beer. Some of them had some of that Brett coming through, and I think a couple were not oxidized and actually tasted okay, which is pretty mind-bending for, beers that are 20, 30, 40 years old. So just to provide some proof here of, back at what I was saying, we actually did run some of those beer samples on the GC to collect some data on what flavor compounds are in them. And we were able to detect some of those phenols that I describe as like part of the funk of Brett in those beers. So pretty clear evidence, even if we were not able to culture Brett, that there was Brett in those beers. So again, is a Berliner Weisse without Brett a Berliner Weisse? It's kind of like there's two different versions of this thing. Some other things we saw is that you even get some esters that persist, like even in quite old beers, ethyl lactate showed up. So that's a sort of a fruity, coconutty kind of ester. It tends to be a good indicator of aging in mixed fermentation beers. It tends to increase over time in bottles. And that's because it's a result of yeast forming an ester from lactic acid. So a sour beer will have lactic acid. There's yeast that can turn that into an ester, and usually that increases over time. So ethyl lactate's a pretty good indicator of age in beers. We saw some other stuff too, but that's sort of the takeaway here is like analytically, even if the beers had no Brett in them, we see some evidence that there was Brett in those beers. So that's not really a debate. Berliner Weisse pretty much always was a Brett beer. And if you look at the, some of the science that was done on the topic, that's true as well. Like if they took the Brett out, the beer was not as good, seemed to be the consensus. We have another past webinar also with our friend Benedict. If you want a little bit more on the history of Berliner Weisse, highly recommend checking that out because I think that's just like a full hour on Berliner Weisse. But yeah, we were able to get yeast out and actually like staggering diversity for these old bottles. So this is a plate, an agar plate from one of those old bottles of Berliner Weisse where we saw many different colonies. And in fact, we were able to isolate several different types of Brett from this old bottle. That's what's really cool about Brett is it can survive in wine and beer for a really long time, decades. So you can actually take a yeast, a sample from the bottom of a bottle of wine or beer, and sometimes if it had Brett in it, that Brett's still alive, and you can kind of culture up something that existed in a fermentation really long time ago. I think people have gone up to 100 years or so with beers and wines finding viable Brett. So that's what's really cool about Brett is it's a survivor. It will probably outlive us if I had to make a guess. It's incredibly hardy, especially in this like low nutrient fermentation environment like finished beer, finished wine. So really cool. That what that means for us is that we can pull strains out of really old bottles and figure out what they can do and see if there's use in modern brewing. So we were able to make a collection. We have a collection of strains from Berliner Weisses. From a number of different species. So the primary species we find in beer is Brettanomyces, which that's what Brett C falls into. Those tend to be the yeasts that are poorer at fermenting, but they tend to have stronger enzyme activity for biotransforming. We also found lots of Brettanomyces bruxellensis, so that's the most common species found in brewing. That's our sort of more standard Brett. Some of those strains are capable of primary fermentation. They tend to be higher producers of those funky phenols. They also tend to be higher producers of the fruity esters as well. So just like flavor powerhouses across the board. And then we also did find some bacteria. So even in some of these old bottles, if it's 20 years old, you can sometimes pick out the bacteria as well, which is pretty cool. So what is it? That was the first question we asked, and that's where we've been doing a little bit of research. There's been a lot of work done in the last few years, and this is sort of part of this update on yeast genetics, yeast genomic analysis. It used to be really, really expensive to sequence the whole genome of a yeast, and that price has gone down, the complexity has gone down. You can use like off-the-shelf analytical pipelines to make it a lot easier. So a lot of that has just gotten a lot more accessible, so that's why you're seeing a lot more genetics work from escarpment labs as well. This has gotten a lot easier and lower cost. But here's an example. Here's just a quick family tree of some Brett strains that I chose because I could find sequence data publicly. Just to show that, just like with Saccharomyces, you might have seen us share the family tree of brewing yeast. Brett has a family tree as well, with typically strains that are closely related in function, are closely related in genetics as well. So you can see at the top, for example, the beer strains are kind of off on their own. They're distinct. They're related to each other more closely than the other strains. You see some of the wine strains there, but actually, a lambic strain in the mix. So there is always some crossover between, types of fermentation, just like we've seen with Saccharomyces. You see kombucha, you see wine. If I added more strains in, there's also some Bretts that are specific to tequila and other fermentation environments. So we were able to take some Brett strains that we had isolated from Berliner Weisse, as well as some of our own Brett strains as well, and do some whole genome sequencing on those and try to figure out where they fit in. Unfortunately, we don't have enough sequences, and we haven't had enough time to really dig into this yet. But we've been able to at least see that these are likely related to each other. At least if we look at this Vilner strain and this Schultheiss strain, both were Berliner Weisse breweries back in the day. You can see that they likely share a common ancestor there, and that they're a little bit distinct from the lambic strains or the Belgian beer strains. So a lot of work to do there, but some evidence that the Berliner Weisse strains are potentially unique genetically, which would make sense, right? Like these things were being used basically in one city for at least 100 years. So there's probably some opportunity. And in this case, like repitched pretty regularly, a lot more regularly than lambic, where your repitching is basically, what drips in from the cool ship, right? In the case of Berliner Weisse, they're actually taking slurry and re-inoculating the next batch. So you would probably see more evolution of the yeast in that, time that Berliner Weisse had its heyday. And you might also see, successful breweries probably passing around yeast culture. There was a little bit of mixing and trading that happened, as well as some consolidation of the businesses later on. So you should see some of these strains be related to each other, and I'm hoping we have a little bit more data. But certainly, we'll get into the flavor as well. But we do see some unique flavor traits of Brett's from Berliner Weisse that, we pulled out of really old bottles. So I just wanted to cover one experiment we did. This feels like ancient times now because it's like pre-COVID, but we did a little project where we made a basic Berliner Weisse. We fermented it with Kolsch yeast and bacteria. Basically made a base sour beer with no Brett, and then we added different Bretts to it and did a secondary ferment in the bottle. So we were able to do some sensory as well as some GC analysis as well. This was actually pretty fresh. I think we had some time pressures for that project. So we probably wouldn't get the full Brett expression after 1 month, but we were able to wrap up this project and get some data at the very least, which I'll take that as a win most of the time with research. Because we wanted to know in this case, like, what is the difference between these Berliner Weisse Bretts? Do they offer something unique compared to the Bretts that we're already offering or that are already on the market? Like, you might hear about, Brett B and Brett C and Brett D and etc. The whole alphabet. We wanted to know, is there something unique here? Because then maybe we can help increase the diversity of Brett beers that are on the market and available to brewers. So what we found across the board is that there's often lower esters in those Berliner Weisse strains. This is just testing 3 strains, but that tends to be true across the board. We also tended to see lower organic acids in the Berliner Weisse strains, which is quite interesting because that might also correspond to funk. In general, having lower organic acids tends to mean that you're going to have a less sort of cheesy, funky beer. So if you really don't like that goat cheese character in Brett, highly recommend a Berliner Weisse strain because they're going to make less of that. Like if you look at the hexanoic acid, the decanoic acid, these are sort of those like goat cheesy characters. You're going to see less of those in a Brett beer. The trade-off is that you're probably going to see slightly lower esters as well. Overall, you're going to see probably a milder aroma profile compared to like a standard Brett B or Brett D. And we did sensory as well. Apologies, the text is tiny here. Didn't see a whole lot of clear links. It's hard to get statistically robust sensory data, unfortunately. But one thing that we saw come up repeatedly was that the tasters noted white wine as a descriptor for Berliner Weisse strains. And that does seem to be the case for, going into the history as well. That seemed to be a desired trait in Berliner Weisse beers, right? This beer is being positioned as the champagne of the North, so it should have a white wine-like character. And indeed, if you make a Berliner Weisse with these strains, you do get that slight sort of wine-like note that helps sort of give that perception of, champagne, for example. We also tried it out in some other beer styles. So this is just an example of a grisette, a non-sour, just straight up fermented with Saison yeast, and then finished with different Brett strains. And then in this case, we were able to do our sensory analysis and monitor it over time and see how do these beers evolve, which was quite interesting. So because we wanted to know, what can, for example, a Berliner Weisse strain do if it's put into a Saison? Does it offer something of benefit here? And the answer is, yeah, I think so. After 11 months of aging, the one with the Berliner Weisse strain, so this is the Hochschule over here. We also tested our Brett B and our Brussels, which are some of our standard Brett strains. It did have more of that pineapple note that you can sort of see at the bottom right than the Brett B and the Brussels. So some nice data on persistence of flavor with the Berliner Weisse strain. Even though it wasn't quite as intense, from the get-go, you see stronger, for example, apple notes with the Brett B early on, but a lot of that does tame down over time during the bottle aging. Okay, so that's it for Berliner Weisse Brett. Always happy to answer any questions. If you do have any questions, if you're checking in live, feel free to ask at the side of your screen. That's helpful for us at the end of the talk as well. So I'm going to talk next about all the things we've tried to make Brett ferment faster and ask the question of, is it even possible? Is it possible? There's a lot of things that have been tried. I won't get into all the details, but there's a lot of reasons to accelerate Brett because it is an industrially useful organism. It has incredible stress tolerance. It can handle acids that other yeast cannot. It can handle all sorts of challenging conditions, but the limitation is that it's a slow fermenter. So there's been a lot of interest in figuring out how to make Brett ferment faster. And same thing for us, right? Like it would be great to have this flavor powerhouse of a yeast that could complete a fermentation like a normal yeast does, rather than taking 4 weeks and requiring a lot of babysitting. So we've tried a few different things internally. We've tried adding glycerol, we've tried adding acetic acid. I won't spare you the details, but those don't work. You can try; they don't work. Some things that work with caveat is increasing the temperature. If you ferment hotter, it will be faster, but it tends to create a trade-off of off flavors. I find when Brett gets too hot, it makes more sulfitic, like vegetable, kind of off flavors. And it can also make more of those organic acids as well. So more of that sort of cheesy characteristics. So that's kind of a do at your own risk situation, going too hot on Brett ferments. So what else can we do? Here's one idea that we thought of and tested out, which I'll call the reverse primary fermentation, or really it's just a blend. But rather than the normal way that we handle Brett, where often it's a primary pitch of Saccharomyces and then a little sprinkling of Brett, like that's a very traditional way to make saisons and all sorts of mixed fermentation Brett beers. We sort of flipped the ratio and tested high ratios of Brett to Saccharomyces. So, from 100% Brett to 100% Saccharomyces yeast. And when you look at the fermentation, you actually see, based on the ratio, the fermentation rate changes, which is pretty cool. So, for example, if you look at the graph on the left, the 100% Brett deferment looks about like 100% Brett ferment. It's pretty slow. The Cali ferment is pretty fast. You can see, in this case, it's done around, probably day 12. Could probably be faster in this is like a lab scale ferment. If it was in a normal size ferment, it'd probably be a little bit faster. But you see, based on the ratio between Brett and Saccharomyces yeast, you do see different rates. So it was interesting for us to see that we can actually dial in the speed of the fermentation based on the yeast originally added. The thought was that the sac is just one of the thoughts was that the sac might just outcompete the Brett completely every single time, and the fermentation curves would look identical, but that's not really how it played out. And what we also found is that the flavor was fairly proportional too. So if you do 100% Brett D, you tend to get 100% Brett D primary flavor. But if you do the different ratios, you get a different mix of that Brett character to that more neutral Saccharomyces character. So that's how we ended up with what we call Optimus primary. The basic idea is that it's a high inoculation rate of Brett and a low inoculation rate of Saccharomyces. And we're basically creating a compromise where we get flavor in the middle between the primary and secondary ferment. We also get a more efficient fermentation rate, faster turnaround, all that stuff that is important to breweries. So that's an option that can be used. We're not able to supply it on the homebrew side because blending Brett and Cali in a single pouch is usually going to cause a little bit of instability or bloating in that little liquid pouch. But you can also do it at home too. You can grow up some Brett D, like build up a big starter of Brett D and basically just pitch like a dash of your house yeast or dry yeast. And that can work really, really well to get you a more efficient Brett ferment. Other things we've looked at to speed up Brett is just diving into their nitrogen requirements. So I brought up the kind of interesting piece that Brett will import nitrate, which isn't typically true for Saccharomyces during fermentation. So it will bring in nitrate. It will use that to generate that really important enzyme cofactor, and then break it down to in that case ammonia that it can also use to make amino acids. So it kind of does double duty there, which is pretty cool. It also has preferred amino acids. So this is true for Saccharomyces. It has amino acids it really, really likes. Which luckily for us, they're pretty similar between Brett and Sac. Like they do tend to like aspartate, glutamine, and glutamate, but they'll use other amino acids as well. Just depends on like how are these building blocks structured, and how easy is it for the yeast to work with these little, protein Lego pieces and build them into proteins. What's kind of quirky about Brett is that it has different preferences based on its environment. So, going back to that Custer's effect of, okay, Brett can, do certain things in oxygen and do certain things without oxygen, and then in between it struggles. It's kind of similar with the nutrition environment. So when Brett's in oxygen, for example, it prefers glutamine, but when it's not in oxygen, that's what the nitrogen here means. It prefers a wider range of amino acids. So little bit complex, a little bit finicky. So the logical thing would be, let's add glutamine and our Brett will ferment faster, right? I'm going to jump around a little bit. Sorry. That's here. So we tried that, testing different amino acid additions with the Brett. And there was, surprise, surprise, because it's Brettanomyces, there was no consistency. So some strains fermented a little bit faster. So that's just release of CO2 in these graphs. Some strains fermented faster when amino acids were added. Some strains fermented slower when amino acids were added. For certain strains, there were some interesting connections, like the aspartate seemed to help Brett D. Glutamine seemed to help Brett Q. So that was somewhat of a dead end. We couldn't add a specific amino acid and boost the Brett fermentation, unfortunately. I'm just jumping around a little bit. So another thing we thought to try, because we've seen anecdotes about this, is hops. We've seen a lot of anecdotes out there, especially in communities like Milk the Funk, that hops can impact Brett fermentation performance as well as intensity of funk. Like there is sort of some common wisdom out there that more hops give you more funk. So thought there was, okay, maybe there's something in hops that's potentially accelerating our Brett fermentation. So we just tried it out. We basically made a concentrated hop tea by extracting hops in boiling water and then filtering that out. So we just had this like hop concentrate, and then we added different amounts of this hop concentrate to a Brett fermentation to see the impact it would have on both fermentation and growth. So we're looking at here in the graph is the growth of the Brett, and you can see it does respond to the hop concentrations. So something about the hops is proportionally increasing the Brett growth. So there's a pro tip you can take home if you want faster and better Brett starters. Get a little bit of hops in there. That does at least in terms of this experiment, does seem to work, and we were able to repeat that. So it does seem to be true. We also tested that in fermentation. So exact same thing, testing the yeast Brett grown with those different rates of hops in fermentation of just standard beer wort. And once again, we did see that the fermentation rate was proportional to the amount of hops added effectively through that hop extract. So there's a take home. You actually can increase the fermentation rate of Brett through your hops. It actually does make a big difference. A wort with no hops is going to likely ferment slower than a wort that is heavily hopped. So, a Brett IPA is probably going to be easier to ferment than like a 5 IBU wort that's being used for a mixed ferment base that doesn't have a whole lot of hopping rate. So just something to keep in mind that, you might be able to find a performance boost in an unexpected place like hops. Why is that? Are hops providing nitrogen? Maybe. There actually is some evidence that you get a little bit of amino nitrogen from hops, but also there is some evidence that hops can provide nitrate. It's one of the few sources of nitrate in beer wort. Can be extracted from hops, so that is a possible explanation. Other sources of nitrate include grape skins. You just might get it from other vegetable matter, essentially. So that's our best theory. We haven't really had a chance to fully dive into that, but we suspect that there's probably extraction of some kind of nitrogen, probably nitrate from hops that's impacting Brett performance, and results in some of the differences that we see here. Which again it's practical for brewing. You can know, okay, if I use more hops, I'll probably get more a faster fermentation. The figures are a little bit misleading. I just wanted to bounce back for a second. The equivalent here is actually 10, like at that highest concentration, it's 10 grams of hops per 10 liters. So 1 gram per liter, not a huge hopping rate to make that difference. We started to look at some other ways that we could supply nutrition to the Brett because we're also looking at amino acids. Okay, individual amino acids don't work very well, but what if we look at a mix of amino acids? Is there somewhere in nature where we could find a rich source of the amino acids that Brett prefers, like our asparagine, glutamic acid, glutamine, those kinds of things? And so in parallel, we were already doing a lot of work with koji. Koji fungus that's used to make traditional rice wines in Asia, like sake. We have a koji product, like it's a dried koji rice that you can buy to play around with making your own miso or sake or shoyu, soy sauce. A lot of different applications there. But what's cool about koji is it's an absolute enzyme powerhouse, and you can make malt with it. So as opposed to traditional malting where you germinate the grain and use the enzymes of the germination process to convert that grain starch into sugar later on during mashing, koji, the fungus, will grow on the outside of the grain. It will colonize the grain and it will secrete enzymes. So protease and amylase enzymes that function similarly to the malting process and break down that malt as well when it's heated or mashed. So that's basically how sake works. You add koji to rice and let it grow, and then make a mash of that, and it converts, and the yeast ferments all that converted sugar into alcohol. So that was sort of the inspiration here. We were playing around with koji barley. It also just smells really, really good. If you inoculate koji on barley, it has this like beautiful lemon, curd, vanilla kind of character. One of the coolest microbial smells, I think, if you're into that kind of thing. I am. So when we thought, okay, we know that koji is also a really rich way to get glutamic acid, right? That glutamate, that umami taste that all the things that koji makes, like soy sauce and miso, even sake, are known to be rich in. It's that sort of savory umami character that a lot of people love. We knew that koji made a lot of glutamic acid, so we thought, okay, let's actually just measure this and see what amino acids pop up when we make wort with koji barley. And it turns out the answer is a lot. We actually got pretty high, these are all just relative numbers here, but pretty high relative rates of glutamine, for example, and glutamate, glutamic acid, specifically when we make the koji wort. Although there are some trade-offs. Some of the other amino acids are lower in the koji wort versus a standard barley wort. But that was cool. Okay, we thought maybe this could be used to grow or ferment Brett a little bit better. And the answer is maybe. So there's just a picture of what barley koji looks like. So it almost looks, well, I mean, it's moldy grain. In this case, we dried it, but really, really tasty moldy grain. So just taking, in this case, hull-less pearl barley, steaming it, inoculating it with koji, letting the koji grow, and then drying it back out. Overall, you can see here on the bottom left, the koji produces a ton of amino acids. So for any brewers who are playing around with koji, keep that in mind. You're going to get a lot of free amino nitrogen from it. It's going to be, superfood for your yeast. And we did try doing some growth. So we actually tested different ratios of a barley wort with koji. And in fact, we did see that it grew better. So that was kind of a cool finding. In some cases, almost 40 or 50% more cells when they're grown in koji. So there's some upstream complexity. I will say we don't do this in production because then we would have to keep making koji malt for ourselves. But it does work. It's something worth playing around with, especially as people are getting more into this sort of crossover between different beverage types. I've seen a lot of people experimenting with incorporating some of the techniques of rice wine like sake into beer, and even vice versa. So keep that in mind. There's definitely some potential there. If you want to make a Brett sake, then certainly I can tell you the Brett will be happy. And certainly it does grow better. So that's some indication that if we give it a mix of those amino acids, it can grow better and probably ferment better as well. So that's that. Not a whole lot of answers. The general point here is give the yeast enough nutrition, and that comes both in the form of amino acids, specifically glutamate, glutamine. And also keep in mind that there's some things that will make Brett grow and ferment better that don't work for Saccharomyces. So hops is one example. Hops are actually slightly inhibitory to most yeast, but in the case of Brett, it seems like hops actually make them go faster. So that's one tool you can use to control your Brett beers. Hoppier beer should ferment faster. So then getting into flavor, this is the other side is, okay, we can make it ferment a little bit faster. What can we do for flavor? Is it possible to control flavor? What ideas can we test there? So I've got a little bit of recent research on that topic as well. So can we optimize for funk? This is kind of an interesting question. We did some work with a client, Little Beast Brewing, out in Whitby, Ontario. They had a really interesting request. They were looking for a unique blend of Brett and Sack because they were making saisons, but they had some limitations with equipment. So ordinarily, when you're making Belgian-style saisons, French-style saisons, you're probably going to be doing more of a Franco-Belgian style wort production where there's usually a step mash involved, which is really, really great to be able to sort of break down the malt and get a really dialed in like nitrogen and sugar profile. But in this case, this brew house couldn't do that. So there's no step mashing possible. But they were also looking to maximize their ester and their phenol production and get that out as fast as possible. So a big challenge, because in the brew house, if you can step mash, do protein rest, do the ferulic acid rest, you can free up the amino acids and the phenol precursors to maximize those esters in that funk. If you can't do that, then you got to be a little bit more creative. So first thing we did was just try to combine different strains. I think this is really cool illustration of if you combine different strains of Sack and Brett, you can almost always get unique combinations. So this is sensory data of just combining 4 different primary Saccharomyces strains in our catalog or back catalog with three of our kind of high funk Brett strains and looking at some of those differences. So you do see some trends and you do see some sort of interesting ones pop out. In this case, we're sort of differentiating between like the medicinal phenol and like the funk. So really, negative versus positive phenol. Funk being more of like the smoke, the barnyard versus the like antiseptic adhesive kind of phenol. So you can see some actually produce more of that and less of the good funk. Some produce more of the good funk and less of the phenol. And then we're also looking for fruitiness too. So keeping in mind the initial idea was how do we maximize both fruitiness and funk with a combination? Maybe we can narrow the field and then tinker from there. So take home from this is like, feel free to, you should play around with different combinations of your yeasts. Your primary yeast that you use in a Brett beer or a mixed ferm beer, absolutely it's going to make a difference in the final results. So it's always worth playing around with it. If you've been making mixed ferment beers and using the same primary strain for years, I recommend playing around with those and seeing if you get different results. You might be surprised. And even some of the new strains out there, like I would recommend, a strain like Thial Libre that's going to bring out a lot of its own flavor, can pair really well in this kind of context. Okay, so we found some combinations that we think might be nice. What can we do to maximize the esters and maximize the funk? So we thought, okay, we know that Brett ferments faster and tends to be more metabolically active when it has more nutrients. And luckily for us, we also offer a nutrient product that we know really, really well, which is our yeast lightning. So we designed an experiment where we basically tested the impact of yeast lightning on Brett to see if this is a way to accelerate ester and phenol production, beneficial ester and phenol production, without having to make adjustments in mashing of the beer. So this is just sort of like a homebrew size experiment, pretty standard saison recipe, testing yeast lightning at different rates. So without it, at the standard dosing rate at double, we picked the Sack strain that the tech doing the experiment liked the most, which is a cool strain. It's a strain just isolated from Blaouise, which is a traditional Saison producer. And then we also used our Brett M and our Q. So we kind of just doubled up to try to maximize the funk. And we did those in bottle all the way through to bottling. And that seems to work. So this is kind of a cool finding. The amount of perceived ester and phenol. I'm not sure if I have the sensory data in here, but analytically, with the higher rates of the yeast lightning nutrient, we would get more esters and more phenols. It was fairly proportional in that beer. So adding the yeast lightning in this case, that's not the primary, reason most people use yeast lightning, but it's an extra bonus that not only is it going to help you with performance in your hazy IPAs, but it's also going to help you bring more flavor from your Brett. So really cool to see that. You can see those ethyl esters, those kind of tropical fruit aromas going up from about 11 times the sensory threshold to about 15 times. So pretty good improvement there. And same thing with the phenols too. So those 4-ethyl guaiacol, 4-ethyl phenol, small increase in the 4-ethyl phenol, which is that sort of medicinal note, but a bigger increase in the 4-ethyl guaiacol, which is that beneficial kind of smoky bacon note. Cool. So I'm going to get on my last session of this little Brett deep dive. So there's been some other works done by other people. I've shown just some fractions of research because, unfortunately for us on the industrial side, we can often only do fractions of research. We're often just trying to answer a question and move on and find a solution for a customer. But luckily, there's also people on the academic side that can do these really in-depth research projects as well. So I do want to highlight one awesome study that was done by a team out of Carlsberg, because in one paper, they were really able to help move things forward quite a lot with our understanding of Brettanomyces in brewing. So, large shout out to the team at Carlsberg who got that work done. So they made a family tree. So I talked a little bit earlier about the family tree of Saccharomyces. Now there's one for Brett, which is really cool. They actually included multiple species. And you actually see that just like what we saw in that really, really basic one that I showed earlier, the beer strains are related to each other. The wine strains are related to each other. They form separate groups. So just like with our Saccharomyces, the yeast has evolved to fit our human fermentation niches. And evolved to be successful and competitive in those environments. And you see that here too. You actually have, lambic Bretts are related to each other. Farmhouse Bretts, which I think is just Belgian beer that's not lambic, also related to each other. Wine Bretts are related to each other. It's a very similar story. You have yeast that adapt to their niches and figure out how to ferment beer or how to ferment wine or tequila as well. They also did a map of the genome of Brett, which looks boring, but I think is actually really exciting because it gives us a lot of useful information as to where fermentation-related genes are in Brett, and then also opens up the door to engineering later on. So now that bioengineering, manipulating the genome of yeast is becoming a lot more common in the beer industry, especially in North America, like here in Canada and in the US. The door is open to these kinds of things. So, maybe it's possible to go in and play around with the genomes of the Brett to make it more flocculent or more fruity, for example. Could be quite interesting. And you need a annotated genome, a map of the genome. You need to know where the genes are and what their sequences are to be able to do any of that work. So, okay, yeah, one other idea. You could screen for which Brett strains have those beta-glucosidase enzymes that help you release bound flavors from hops and fruit using the genomics. And you could, either plug and play that gene from one Brett strain into a more efficient one, or understand, what version of the gene does a really efficient strain have. And what's really cool here is that genetics keeps getting cheaper. Unfortunately, lab techs to do the bench work does not. So anything we can do to use genetics to do as much of the work for us as possible, like basically do the sequencing to get the raw data and then use computers to understand that data, is the better. And I think as a result of this, you're going to see yeast develop a lot faster in terms of new strains and engineering and new, ultimately for you, new flavor and efficiency possibilities. So who knows? Maybe someone's going to figure out how to make Brett faster this way. I'm not sure. Another cool thing they did is that they actually correlated growth in cellobiose, so a wood sugar. That's a sugar found, a carbohydrate often found in oak barrels, for example. They correlated that with beta-glucosidase activity. So the Brett strains that have better growth in cellobiose often also have better beta-glucosidase activity. So this could be a way to do strain evolution, right? You could grow a yeast in a high cellobiose environment and potentially get more beta-glucosidase activity. Can neither confirm nor deny that that's possible, or you could just use it to screen for biotransformation, right? Measuring beta-glucosidase is not super difficult. It's not measuring thiols or anything like that, but growing the yeast in cellobiose is quite simple. You can just basically set it up in a sort of a liquid culture and screen it. So it would be a good way to screen a bunch of Brett strains, maybe go back and screen our Brett library for all sorts of beneficial traits like the beta-glucosidase, which it's really relevant, right? A yeast with high beta-glucosidase activity, like Brett C is the classic one, is going to typically produce more aroma because it's going to release some of those bound flavors from malt. Sorry, not from malt, from hops, and also from fruit. Like there's even been some work done by the Belgians showing beta-glucosidase release of flavors in cherries, for example, or coriander. Another really cool thing they found was they actually found a Brett strain with no 4-EP phenol. So that's really cool because it's almost like a non-phenolic Brett, if you can imagine such a thing. I think Carlsberg has done some work to try to patent that because they did discover it. So that opens up a really interesting flavor possibility in the future as well. You could potentially have Brett beers without the phenols, and it's kind of, for me, hard to imagine what that would taste like, but I think it could be really interesting, knowing that Brett is such a flavor powerhouse in terms of esters and in terms of biotransformation. If we're able to remove the phenols, we could open up the type of beers that can be made with Brett. Probably have a little bit more application in IPAs, for example. So that could be really interesting as a technology development in the future for the world of Brettanomyces. These guys did a lot. They also looked at differences in terpene biotransformation between both primary and secondary fermentations by Brett. So it's hard to see because the scales of the graphs are different. But you do see that terpene conversion seems to be a little bit weaker in secondary fermentations. So our team has done a lot of research on Brett primary fermentation and some of the flavor potentials there, but we were never able to look at terpenes, which are a really important class of compounds that produce a lot of those floral or citrusy or even fruity and mango-like aromas in beer from hops. So once again, it does seem like you get a little bit more flavor potential from the primary ferment versus a secondary ferment. So that's important to keep in mind, right? There's these trade-offs. Brett primary fermentation is going to be really flavorful, but it's also going to be frustratingly inefficient. And then beyond beer, there's a lot of cool things that can be done with Brett, and I want us, hopefully to think and be a little inspired to think outside of beer, especially as breweries are making a lot of different types of products. There's been some really cool research done on Brett's role in kombucha. It's actually one of the primary yeasts found in kombucha because it's so good at surviving in that environment, specifically in handling high acetic acid, high vinegar sort of content. Brett is an absolute champ. So you often see it show up in kombucha. And so now that that's been understood and a lot of the mystery of kombucha has been removed, there's actually some work to develop this and improve it. So one example is this study where they combine different strains of Brettanomyces bruxellensis and Kamagatabacter, if I said that right. It's one of the bacteria found in kombucha that makes that like SCOBY pellicle. Just those two things alone was enough to make a full kombucha fermentation with that pellicle or biofilm on top. So kind of opening up the world of like pure culture kombucha. You know, we think of it as a mixed culture thing where we've got a SCOBY and it's just got some different microbes in it. We don't really know what's going on. In the future, it can probably be dialed in and made very, very specifically with specific bacteria and yeast. And that opens up the possibility of actually dialing in flavors in kombucha in a way that we already do in wine and cider and beer and almost everything else. So one potential place where Brett strains might find a lot of utility. Cool. So that's about it for me. That I was hoping to provide a good update on where things are at with Brett, and keep you informed that we're still moving and trying to keep things exciting with Brett and make these easier to work with, because that's one of the challenges is the learning curve with Brett is crazy. So hopefully that was helpful if you're tuning in. I want to give acknowledgement to some of the researchers who helped produce the data used and shared here. So Phil Jurek, who's just left our team and moved back to BC, did a lot of great work as an R&D tech, testing the impact of yeast lightning on funk, and also just producing a ton of Brett beers in-house here that I've had the pleasure to drink. And then also a lot of people in the history of Escarpment Labs over time. We've been studying Brett basically since the start. So a lot of people have been involved in Brett research that I've shared here. So Iz Neto, Caroline Tarawa, Sarah Trainer, and Brianne Schooley. A bunch of people that have been involved in researching Brett and helping us spread the gospel of our funky friend. If you want to know more, and in fact, there is more. Keep this in mind. We have almost 2 entire more webinars on Brett and some stuff in our blog and our knowledge base. So it's a passion of mine that should come across as clear. But if you're making Brett beers or mixed ferment beers and you want more information, you should be able to find it on our resources. So you can check out the blog on escarpmentlabs.com. You can check out our YouTube channel, where you can find out a lot of different things about fermentation, as well as our knowledge base, where you're going to find more tailored fermentation advice and deep knowledge. So check out all of those things. They're all useful resources for you as a brewer. And that is it for me. Thank you for tuning in live or checking us out on YouTube. We have a few more webinars coming up as part of this series before we'll take a little bit of a break, because I need to come up with some new ideas. So next week, we're going to be talking about beer yeast nutrition. Following week, we're going to be talking about non-alcoholic beers, which is a very hot topic this year. After that, 2 weeks after that, we're going to be talking about controlling beer off flavors. So you can also check out our blog, because Nate Ferguson from our team has been starting to release some blog posts as well about controlling beer off flavors. And so I'll be putting together a lot of that stuff and sharing it here. And then finally, we're going to be talking about using beer yeast in cider, seltzer, sake, wine, and mead. So all the different places that you can use beer yeast, because regardless of what it's fermenting, beer yeast has a lot of advantages that we can make use of. We actually just made a cider with our house ale dry yeast, and it was done fermenting in 7 days. Very easy to crash, carbonate, tastes delicious, very, very clean. So there's one example right there. We'll get into some other options as well. Okay, I'm just going to check into our questions. If you have any questions, if you made it to the end, feel free to go into that Q&A box and ask a question. But I think I'll get on to a couple here. Okay, so I got one from Travis. Travis could not attend, but he's got 2 questions. One, I know that 100% Brett fermentations tend to not attenuate as much as mixed ferments. However, if trying to do a 100% Brett ferment, is sac contamination a concern? Will a small amount of leftover sac pose risk for bottle bombs? I think it depends on the yeast. What I will say is that sac contaminations are very common in 100% Brett ferments. If you have a 100% Brett ferment that's done fermenting in 7 days, it's either contaminated or you've figured something out that science hasn't. So it's always worth checking under the microscope if you've got one. See if you see those big round Saccharomyces cells, or if it is in fact all Brett in the mix. In terms of bottle bombs, I think that's still a bit of a knowledge gap. We don't actually know how far some Brett strains will ferment in all environments. It does seem like 100% Brett ferments tend to not dry out beer as much as mixed ferments do, and certainly not as much as diastatic yeast does, like saison yeast. So that's an area where I would be concerned. If you have 100% Brett beer, I find those tend to end in like a normal wort. Those tend to end around 1.008 or 2 Plato most of the time. So if it were contaminated with a saison yeast that's going to ferment down to 1.002, then yeah, you have a risk of bottle bombs. So um, just something to keep in mind. Again, I would say for 100% Brett beer, if you're home brewing and you're also using saison yeast, where there is that potential of cross contamination, I would even just like keg the 100% Brett beers and bottle the saisons if you're worried about bottle bombs. The other thing is just keep an eye on it, right? And if it starts to overcarbonate, make sure that all that beer gets cooled down. He's got a part two. I've noticed when I do Brett starters, they end up smelling very fruity, and I attribute this to increased oxygen exposure. How far into the fermentation can I push additional oxygen to a Brett fermentation before things start to turn acidic? Will Brett only start synthesizing acetic acid at terminal gravity? Thanks. No, it will start synthesizing acetic acid sooner. So really your control here is alcohol. So as soon as your specific gravity is starting to drop significantly, that's when you probably don't want oxygen. So you absolutely can oxygenate a Brett ferment a few times early on. I have even recommended that for primary ferments. It will work to sort of speed up that lag phase. You can oxygenate the Brett, day 1, day 2, day 3 kind of thing. As soon as you have alcohol is when things can get messy, both in terms of acetic acid and also in terms of THP, which is that sort of Cheerios flavor that you can also get as an off flavor from Brett. But both of those are dependent on ethanol. So once your alcoholic fermentation is really started, i.e., the yeast is done growing, you have a fermentation where you can measure that specific gravity drop, that's when I would avoid oxygen. But you're right, on a stir plate, you're going to get a pretty, nice fruity Brett ferment because you're getting a lot of growth, right? And you're also getting some fermentation. But I would just be, a little bit cautious there. Okay. Another one from Jeffrey. How much concern is there for contamination if someone uses Brett and Sac at home? Will Star San or IO Star both take care of it? Some, I would say across the board as a contaminant, diastatic yeast, saison yeast, is way scarier than Brett. Some may disagree with me. Depends on what you're making as well. If you're a winery, then Brett is public enemy number one. If you're a brewery or a home brewer, then I would say diastaticus is public enemy number one because it can cause so many problems like exploding bottles and off flavors and stuff like that if it gets in the wrong place. Brett is hard to kill if it's in the wrong place. So it also depends on what equipment you're using. Like if you're fermenting in plastic and you're using Brett, then just assume that you're always going to have Brett in that fermenter forever, right? Because it's probably just going to survive. If you've got like a stainless steel fermenter that you can like really, really clean and really, really kill with heat, then it's a lot less of a risk. So it all kind of depends on what you're working with. In terms of sanitizers for killing yeast, I would like on a homebrew scale, I would recommend an iodine-based sanitizer. Tends to work really, really well to kill yeast. Those are not as fast as something like Star San, but they will often be more effective. So if you have a piece of equipment and you're worried about, Brett cross-contaminating from that equipment to something else, a soak in an iodine-based sanitizer would be my recommendation because the iodine is really effective at inhibiting yeasts. Sack and Brett blend, co-pitch versus primary and Brett in secondary. What works best? This is from Thibault. What could be the differences in terms of flavor for these two processes? Thanks for the presentation. Thank you. Thank you for tuning in. I will point you to one of our other webinars because you'll get a little bit more information there. So the webinar we have on Brett with Caroline back in 2020, she went over a lot of the work from her thesis at the University of Guelph, where she looked at this exact topic, co-pitch versus primary versus secondary, and did some big experiments to measure the differences. If you're going for flavor, the answer is primary is better. You're going to get the most flavor from primary. Between co-pitch and secondary, there's actually very little difference. So it just really comes down to what's, at least analytically, it comes down to what's convenient for you. Analytically, we were not able to see much of a difference between a co-pitch, like adding both yeasts at the same time, or adding the Brett after the primary ferment. So that just comes down to convenience in a lot of cases. One thing to keep in mind there, though, is that we did see some differences between primary strains. So like there's this thought that Brett's going to totally pave over the flavor of your primary yeast strain, and that doesn't seem to be true. We were able to, the primary strain would always leave its fingerprint on a Brett secondary or co-pitch. Another one from Deej. Anecdotally, it seems like adding lactic acid bacteria to a very barnyardy Brett beer will significantly reduce that barnyard funk. Are those funk compounds locked in once formed, or are they changeable? You might have me stumped there. I'm not sure. I do find that the perception of that funk is more present at beers with higher pH. Like you can have a beer that's analytically higher in phenols, but if it's more acidic, it tends to be perceived less. So there might just be some weirdness with the acidity impacting the flavor perception. I'm not sure. There's probably some research out there on the wine side. Like one example that I can suggest is if you look at wine, Brett can grow in barrel-aged red wines or white wines, but it's a much bigger problem in red wines, typically because, well, first of all, because they tend to have more precursor for phenols, but also because of pH differences. A lot of red wines tend to be higher pH, and you might notice those, this is just kind of spitballing here, you might notice those funky aromas more versus a white wine that's like quite acidic and crisp. That's interesting. Eric, I got a question. You mentioned acetic acid production. If one wanted to make malt or balsamic vinegar, what yeasts would you recommend? Yeah, that's a good one. Kombucha SCOBY is what I would recommend. It's pretty interchangeable with Vinegar Mother. The nice thing with kombucha SCOBY is you can do it all in one. A lot of Vinegar Mothers just have the bacteria, so you still need to make the alcohol in the first place. With a kombucha SCOBY, it's kind of synergistic. You can expose the kombucha SCOBY to a source of sugar, and it will produce, the yeast will produce some alcohol, and then the bacteria will convert that alcohol into acetic acid. So that's a great way to do it. Like Brett on its own is not going to make acetic acid at the level that we would call like actual vinegar. It would just make like a vinegary beer. So if you want to go that far to make like a full malt vinegar or like wine vinegar, I would start with a kombucha SCOBY. They tend to work pretty well. I can say, I play around a lot with food fermentation at home, and I have made plenty of different vinegars using our kombucha SCOBY. And again, the benefit is you don't have to do like the primary, secondary ferment with that. You just basically make kombucha, but instead of bottling it after a week, you leave it for a few more weeks, and then you have vinegar, and it works pretty well. Cool. I think that's about it for the questions. I think I'm going to sign off. I'm hoping to see everyone next week in our talk about beer yeast nutrition, a deep dive. Until then, keep funky.